So this is the walkthrough, the week 3 lab, cell structure and function, cells types, and transport. Make sure you get the right one because they're not quite in the right page number because that's confusing and totally unnecessary. So you're going to complete the document that's formatted and look at the rubric. Let's look at the rubric first. So here's the rubric. Remember, it's due on Sunday. So there's going to be 4 exercises, each a point with the observation being a half a point, and then half a point for the complete sentences. This one, again, you need to have references. And then again, it reminds you, you can draw stuff out, you can scan it in, you can take a picture of it, whatever works for you. For the missing part, just make sure you put it into your lab and not have it as a separate assignment, tab submittal. Okay, now let's go look at the tab. Which one is this one? Cell types and transports. I will upload my key to so I can look at both, so you might get some weird pauses. So put your title page here. Yes, I do like correct title pages. And then we have our whole thing. Observations. Again, this is when you can share it or not share it. Just make sure you say what your organism name is. So which one was your prokaryote, which one's your eukaryote, stick the name in, and then say whatever you saw. Delete anything. Well, say what you saw, right? That's what you're going to see inside of it. Then we're going to include our drawing down here. So take a picture, scan it, whatever works. Or draw it really confusingly, I think. You could do it in Word, but it's going to be a pain in the butt. Then we have our data table 2, which are diffusion rates, so you'll say your times. And then we have our table 3, so what's in it, what's the dialysis tubing, what's in it, what's its initial color, what's its final color, was it positive or negative color. I would say the color and say that's positive, that's negative, and then what's going to end up in it at the end. So hopefully it should have 2 different things, right? At the end, we're probably going to have something that moved through one way or the other. I would guess both ways. So then we talk about results. And they say, what is it? What are the differences observed between the 2 cells? So what did you see that was different? What's the difference between a prokaryote and a eukaryote? And then here we're going to have list the specified cell part for each cell possesses. So escape from hazardous condition. What part of it's going to allow it to do that? Regular water content, what structure does that? Store food, what structure does that? Reproduce. How does it do that, Is it going to be division, asexual, budding, what? Okay? So these are kind of structures more, and these will be, this down here will be process. Okay, this is kind of what were you used to more? It should be the one of most things are around is what you're probably more used to. Let's just answer which type of cell would be most successful in extreme environments. This would be a great one to cite too, because you could say like, oh, I looked it up, or you can say what you think. Whatever works. It's getting late. I've done a lot of videos today, but that's okay. We're gonna plug on through, right? How are you going to see it? That will kind of be a reference back to the week 2 microscopy lab. And we're going to research stuff online, so this is where you definitely have to cite somebody. And then discuss whether cyanobacteria or amoebas are beneficial or detrimental to humans and the environment in general. Kind of pick one. And then we're going to say difference observed again. So this is more on the eukaryotic side of things. Last one was more on the prokaryotic, right? And then, so what do they have? What do they not have? Let's see. I see. Okay, then we're going to have your plant versus your animal, right? So, your plant cell, what does it have to do this? Your animal cell, what does it have to do this? Or is it going to eat something or something? How does it store food? What's the process that we're doing? These should hopefully be different than the previous table because you're looking at other things. You say what you're most familiar with, animal or plant, kind of toss of a coin, right? And then successful in extreme environments, another one you could research or say what you feel. And then we're going to outline the differences between the 2. I would probably do something like, the plants has chloroplast, no chloroplast. This one has whatever, that one doesn't. This one has whatever, that one doesn't. And the similarities, they both have blah blah. They have blah blah blah, kind of thing. Then, which medium exhibited the fastest rate of diffusion? You're going to see how exercise 3, so this is going to be with our dialysis sucker. Our ratios. Graph the two sets, so you should be showing me a graph, right? Again, this is when you might just take a picture. Apparently I can't spell it. Insert. I've already uploaded it, so it's just going to be slightly incorrect in the thing you download, but you can see it's wrong and laugh at your teacher. And the hours of work it took to get this reasonable. The format was so bad. Oh my gosh, seriously, it was so annoying. And something about UOP. Why do you not format everything to be an APA? Okay. So were your two hypotheses supported or refuted? I've not scrolled down to that yet. What letter are we at? A, discussion. Where's discussion? There's discussion. Okay. You say what you got. This one you tested. What was the exercise? Why is this exercise an example of passive transport? If you're not sure, this is a good thing to go look at it. Maybe research passive transport so you can be remembered. You can be remembered, you can be reminded. Do you think it was drinking? And then why must the spread of coloring gelatin be monitored? Why can't you just leave it? What would be a good graphic method to detect the sucker? And construct a pretty table. So insert a picture graph down here. And then finally, this is one I hopefully said and reminded you, I'm pretty sure I did. I said I did, that you'll share this information with your other students in the assignment area so that you guys can have a bigger example to look at. Just helps. And then we have questions. What are your test substances? This one doesn't actually just tell you in the thing, but you know what you put in there. So what is it that you want to know if it moves around? Okay, what moves around? Make sure you do that right. Why did the color in the jar change color? What does that indicate? So when you put a color in there, what are you testing for? That's probably going to be one of your test substances. And the bag, did it change color? What were you testing for? Your other test substance. What does it do? This will be a reminder of the last week's. What did not go through? Why? Was it too big, too small? Weird somehow. What happened to your hypothesis? How is the tubing similar to a plasma membrane? If you're not sure, good place to have an in-text citation. What type of transport mechanism does this exercise stimulate? So we're going to guess it's not passive. And let's see, what was the name of this place again? The name of this section, if I scroll up here, was an example of active transport. Therefore, I will assume that it is an example of active transport. Make sure you read those titles. They're stupid and silly, but a lot of times when you're doing a lab, you just get stressed out and brain dead, and you'll miss that clue, right? So always read the title and be like, what does it tell me? I'm testing this. Okay, this is probably going to be a question too. And then what can you conclude? And then do a lab summary and have your reference section at the end. As always, lab summaries, I think in this one too, they're not required, but if I see one, I will be more lenient. Always remember. And you can submit for a draft review. Draft for this would be due on Sunday by noon. Hopefully, I'll have to figure out my schedule. I think Sunday by noon works. I will let you know. But it's usually noon on the due day, so it should be Sunday at noon. There goes my Sunday. But only in the morning, right? So it'll probably be closer to noon. If you turn it in really early in the morning on a Sunday, I will likely sleep in, so I will get it to you closer to noon. Okay, I hope this helps.